Welcome back. In this video, I will discuss glycolysis. The word glycolysis comes from glycose, which means sugar, and lysis, which means breakdown. So, glycolysis is a series of enzymatic reaction that sequentially oxidize glucose into pyruvate. And during the oxidation process, ATP is also liberated. After a heavy meal, glucose is liberated from starch-containing foods. Glucose is quickly absorbed via villi and finally mixed into the bloodstream. In active cells, especially muscle cells under vigorous exercise, the intercellular glucose concentration is generally low. To maintain osmotic balance, glucose enters inside the cell and the cell quickly phosphorylates it, making it negatively charged. The plasma membrane is also negatively charged, and so glucose 6-phosphate is not able to come outside the cell. It is also chemically changed from glucose to glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. Hexokinase needs energy to drive the reaction in the forward direction. So one molecule of ATP is utilized in this reaction. Hexokinase can phosphorylate other hexose like glucose, mannose, fructose, etc. In the liver, another enzyme called glucokinase is present. It can specifically act on glucose. The main function of glucokinase is to control the blood sugar level. When the cellular ATP pool is nearly exhausted, the second enzyme called phosphoglucoisomerase comes into action. It isomerizes glucose 6-phosphate into fructose 6-phosphate, another hexose. Fructose 6-phosphate is again phosphorylated at the number 1 carbon position with the help of phosphofructokinase. Here again, the enzyme uses the energy of ATP hydrolysis to convert the substrate into a product. The product now contains 2 phosphate groups, one at the 6th position and another at the 1 position. And the product is called fructose 1,6-bisphosphate. Now the 6-carbon compound is cleaved in the middle with the help of aldolase enzyme. 2 3-carbon compounds are liberated as products called glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Look the numbering of carbon atom. It is changed here. Carbon atom 1, 2 and 3 now become atoms number 3, 2 and 1 of dihydroxyacetone phosphate, and the order is reversed. Atom 4, 5 and 6 become atoms 1, 2 and 3 of glyceraldehyde 3-phosphate. An interesting fact is that there are 2 classes of aldolase present. Aldolase 1 that is present in eukaryotic cells, but aldolase 2 that is present solely in prokaryotes. Therefore, aldolase 2 can be an excellent drug target to kill microbes that causes infection. In this reaction, one keto compound and one aldehyde compound are liberated, and the reaction is exactly opposite to aldol condensation. Another interesting thing is that glyceraldehyde 3-phosphate is ready for the next enzymatic reactions, but dihydroxyacetone phosphate is not. Instead of synthesizing separate enzymes for dihydroxyacetone phosphate, cell converts it into glyceraldehyde 3-phosphate. The cell can convert each other with the help of another isomerase called triose phosphate isomerase, or in short, TIM. Now 2 molecules of GAP are prepared. Each molecule can proceed one by one. So remaining reactions will occur 2 times. Once when GAP is entered and another round for GAP also, but it is converted from DAP actually. Both bromohydroxyacetone phosphate and glucidol phosphate can inhibit the catalytic action of TIM. GAP is oxidized and phosphorylated by the help of glyceraldehyde 3-phosphate dehydrogenase. The enzyme uses inorganic phosphate as the phosphate group donor instead of ATP, and 1 molecule of NAD is reduced to NADH. Cysteine is present in the active site of this enzyme, and therefore, if iodoacetate is added, the enzyme will be permanently inhibited. Iodoacetate is called an irreversible inhibitor of this enzyme. Next, 1,3-bisphosphoglycerate donates its 1-carbon phosphate group to ADP, and resulting in 1 molecule of ATP is liberated. Actually, 2 molecules of ATP are produced. You can remember this reaction. I have already mentioned that from now onwards, the enzyme will convert the substrate into the product 2 times. Once for GAP and the second one for DAP. So, after 2 rounds of this reaction, 2 molecules of ATP will be produced. The name of the enzyme is phosphoglycerate kinase. After this reaction, the two ATP that are used in this and the previous reaction are compensated. The net ATP production till now is 0. 3-phosphoglycerate interchanges its phosphate group from number 3 carbon to number 2 carbon, and the enzyme that catalyzes this intermolecular group transfer is called phosphoglycerate mutase. The enzyme and the substrate are very interesting. If you radiolabel the phosphate group, what you can expect? A radiolabel product like this will release, but here the enzyme itself become radiolabel. That means one phosphate group was already present in a bound state that is actually donated to the substrate, and this radiolabel phosphate group is transferred to the enzyme. 2,3-bisphosphoglycerate is also liberated occasionally, and as a result the enzyme becomes inactive. The activity can be restored by the help of a reversible reaction, and some amount of 2,3-bisphosphoglycerate are always present inside the cell. 2,3-bisphosphoglycerate also alter the oxygen binding affinity of hemoglobin in RBC. If hexokinase, the first enzyme, is defective in RBC, that means glycolysis is stopped and no intermediates will produce, including 2,3-bisphosphoglycerate. If pyruvate kinase is defective, the concentration of 2,3-bisphosphoglycerate will increase, and the O2 binding affinity of hemoglobin also changed, as you see in this graph. In order to transport oxygen efficiently, glycolysis plays an important role in RBC. And next, 2-phosphoglycerate is dehydrated to form phosphoenol pyruvate, and one molecule of water is liberated. The name of the enzyme here is enolase. Here the enzyme is called aldolase, and here it is called enolase. And the product in this case is one enol compound. And finally, phosphoenol pyruvate is converted to pyruvate by the help of pyruvate kinase enzyme. And here again the name of the enzyme ends with kinase. That means one phosphate group will be added or may be removed from the substrate. Here the substrate is dephosphorylated and the phosphate group is transferred to ADP. In the last step of the glycolysis, again two molecules of ATP are produced, one from GAP and another from DAP. So at the end, two molecules of ATP is our net profit. Glycolysis is one of the primitive metabolic pathways used by most organisms on this earth, including microbes. And most of the enzymes are conserved throughout the journey of evolution, and the pathway does not need any free oxygen. And therefore, anaerobic organism can also use glycolysis for the production of ATP. All 10 enzymes are present in the cytosol, and they generally present close to the plasma membrane. The entire metabolic pathway was discovered in 1940, and the branch of biochemistry slowly started after the discovery of this pathway. The credit goes to three scientists, Emden, Meyerhoff, and Parnas, and that's why this pathway is also called EMP pathway. The pathway starts with 6-carbon compound, which is glucose, and the final product is 2 molecules of pyruvate. 2 molecules of ATP are utilized in the first 5 steps, and it is called the preparatory phase. And later, 4 molecules of ATP are synthesized. It is called the payoff phase. So the net gain is 2 molecules of ATP and 1 molecule of NADH. The fate of NADH will be discussed later in ETC. So that's all for now. If you like this content, press the like button and make your comments. If you are watching my videos for the first time, press the subscribe and to get regular updates. Finally, thanks for watching.